Hi guys, this is the Bio Kid here, and today we're going to learn about an important topic in biology, that is the Lac Operon. So, operons actually are a set of genes that codes for certain proteins that helps them survive in needful conditions. And keep that in mind that this is controlled by a single promoter. A single promoter's presence ensures that they are all transcribed together. That's how they control the biochemical processes of a cell. Now, some of the operons are tripoperon, that is the tryptophan operon, and also the lac operon, of course, that is the lactose operon. Now, what is lac operon actually? Lac operon is a cluster. It's a cluster of three structure genes that is involved in, lactose metabolism. Lac Z, that there are three genes actually, that is the lac Z, the lac Y, and lac A. The LAC-Z codes for enzyme beta galactosidase. So this enzyme helps in the breakdown of lactose into glucose so that the cells can utilize it. Now LAC-Y, this codes for enzyme permease. It's a membrane protein that helps in direct influx of lactose on the outside. And the LAC-A, about the LAC-A, which codes for lactose transacetylase. Nothing much is known about this yet. Now it is to be noted that upstream to the promoter there is a lac I gene that encodes the repressor protein. Now, when do E. coli use lactose? It's obvious that when glucose is not present, or it's in a minimal level, you know E. coli can use up lactose. But keep in mind that lactose is disaccharide, so it is needed to be broken down or hydrolyzed into glucose, and so glucose is, the preferred solution, or glucose is preferred by the E. coli cells. Now, coming into the mechanism, uh, it's to be noted that a small amount of sugar allolactose is formed within the bacterial cell, which fits into the repressor protein of the allosteric site. You know? uh, let's start with this. When lactose is absent, you know the repressor protein, which is you know which comes from the lac I gene, it sits on the promoter region of the lac operon, as a result of which no gene expression takes place, or no RNA transcription takes place. Now, in condition B, when lactose is present, uh, a molecule of lactose comes and sits on the allosteric site of the um, it's called the repressor protein, as a result of which it falls off, and now the genes can be expressed, or as we should say, RNA can be transcribed. And that's how the lac operon works. Now, conditions for regulation. uh, let's start from the bottom. you know uh, when glucose is absent and sorry, lactose is present and glucose is absent, RNA polymerase sits on the promoter and transcription takes place. That's we have learned, uh, from the previous slide. Now, when, the next two conditions from the bottom, we can see the RNA polymerase, you know, it cannot sit on the promoter, is because, you know, it is blocked by the repressor protein, so no transcription takes place. Regarding the first case, when glucose is also present along with lactose, the RNA polymerase sits but falls off from the promoter. And the reason behind that I will be explain it in my next video of mine. So here also there's no transcription takes place. Okay, so that was it. So stay tuned, and if you like the video, share it with your friends and subscribe to BioKid.